Dr. Sudha from ISER Pune to give a guest lecture on the astrobiological narrative of life's origin. I feel extremely honored to introduce her. Dr. Sudha is an associate professor in biology at ISER Pune. She runs School Lab at ISER Pune, which stands for Chemical Origins of Life. And her research areas are mainly focused on the origins of life and its early evolution. Welcome, ma'am. And I request the participants to put any questions in the chat box and we will take them at the end of the session. And
thank you. Hi, Jaya.
Can
you hear me clear?
Yes, ma'am, it's audible.
Okay, I'm going to go ahead and share my screen. Can you see the screen?
Yes, ma'am.
And you see it in the full mode now?
No, ma'am, it's not in the full mode.
Okay, hang on. I think I know what's happening. Yeah, just give me 1 second. I'm going to stop sharing. Share again. Full screen. Share. Can you see it now?
Yes, ma'am.
Full mode?
Yes, ma'am. Now it's visible.
And did it move?
Yes, ma'am.
Perfect. Okay, so shall we get started?
Yes, ma'am.
Yeah, awesome. Good evening, everyone. First of all, thank you to Spatz, I don't know how you say that, and especially to Jaya for inviting me to this very interesting event, which I didn't know existed, the NSSC 10th edition, which IIT Kharagpur hosts. It's very exciting to know that you folks are doing this, and it is super exciting to sort of appreciate the fact that you think astrobiology is a very important part of this particular realm of understanding space and our space in it, so to speak. Yeah. So today what I'm going to do is give you a sense of what is the astrobiological narrative of life's origin. And what do I mean by astrobiology? Astrobiology is the study of the origins, the evolution, the distribution, and the future of life in the universe. This is sort of a modified definition. Back in the days, the NASA Astrobiology Institute sort of separated the definition from what we call, there was astrobiology and there's exobiology. Astrobiology was predominantly the science that dealt with understanding how the origin and evolution of early life and its distribution happened on Earth. And exobiology was the stuff that was supposed to do that pertain to research that is done on non-Earth exoplanets and other planetary bodies. But over the last, I would say, decade or so, the definition has evolved, and this is because of a lot of the understanding, scientific understanding, that has added to our appreciation of what is it that we know about life. There's a working definition of life that we use when we talk about it, and what might have led to certain planets being able to host life as we understand it, and what might allow for its emergence, its evolution, and its spread across various planetary bodies. So let's get going. As Jaya mentioned, I along with my students run the COOL lab. It's the Chemical Origins of Life lab. And yeah, so this is the group that, the basic sort of core group that sort of has been in the lab for over a decade now. Very first student who joined me, the second student who joined me, they've graduated now. The others are newer graduate students. Actually, these three are seniors and they're going to graduate soon. My postdoc, two other newer graduate students, and subsequent to them, I've had two more join, and a whole bunch of undergrads who have been the reason why we've been able to carry out research in questions that we're interested in, about which I'll touch upon later. But prior to that, I'd like to set the stage and give you a sense of the context that allows us to pursue the questions that we are interested in a very structured manner. So this brings me to this very important story of how we think biomolecules originated. On this particular slide, what you're seeing is this very important continuum that exists between physics, chemistry, and biology. What is physics? Physics is when you're talking about atoms at the atomic level. What is chemistry? A bunch of atoms coming together to form small molecules. And here are two images which are essentially examples of molecules that have been found in space. And on the right side is this very complex molecule called buckyballs, which is made up of about 40 to 60 atoms of carbon. And these kinds of more complex complex organic molecules have also been found in space. Now, when you have these smaller molecules interact with each other and form what we call biomolecules, the molecules that are relevant to life as we understand it, you can see that you have this beautiful continuum that goes from physics to chemistry to biology, which essentially allowed for the transition from non-life to life on the early Earth. So how did they come about on our planet? So there are two hypotheses in this regard. One of it invokes exogenous delivery. Exogenous delivery is when something comes from outside. It's thought that these molecules, which can be very readily formed in space conditions, could have ridden on meteorites or comets. And there was a very early phase in Earth's formation when we were constantly bombarded by these planetary bodies like meteorites. So a lot of these organic molecules, including water, is thought to have ridden in these planetary bodies and is thought to have been brought to Earth. One other hypothesis posits that terrestrial syntheses in certain niches, which facilitate very interesting chemical and thermal fluxes, would have also allowed for us to transition from this inorganic space to organic space, resulting in the formation of several biomolecules, some of which have been listed here, including some amino acids like glycine, alanine, glutamic acid, and other hydroxy acids like formic acid, lactic acid, acetic acid, and so on and so forth. So we have two ways in which biomolecules would have been plausible on Earth, and that would have set the stage for the transition from non-life to life. Now, if we want to understand this transition, what is the best way possible to do that? There should be some good place that you can start working on, and then working your way either using a bottom-up approach to understand the process or a top-down approach. We use a bottom-up approach to understand how the transition from non-life to life might have happened on early Earth. And this slide provides a clue to what is it that allows us to do these sort of studies in a very systematic manner. Here is a slide which shows you various entities, all the way from mushrooms to the giant sequoias. There's a supergiant catfish, a microbe. This is T-Rex, and you also have the very honorable Nelson Mandela. What is it that ties all of these entities together, living entities together? What is the least common denominator? Any guesses? Anyone can just guess, unless and until this is a complete unilogue or a monologue. What do you think ties all of these things, these living beings together?
Genetic material, like all have DNA.
Genetic material. Okay, that's a reasonable answer, but fundamentally, they're all made up of cells. Cells are the most fundamental units of all life, within which the genetic material that you're referring to is present. So one of the good ways to talk about the transition from non-life to life is to try and understand how did you go from that biomolecular space, which I was talking to you about, where the molecules are still existing as very large chemical entities, but then the question is, how did they come together and eventually lead to the emergence of life-like cells? That is sort of the paradigm within which we try to understand this transition from non-life to life. And here is the fundamental unit of life's very famous, two very famous examples. On the left is what is called a prokaryotic cell. On the right is a eukaryotic cell. We are all eukaryotes, right? Prokaryotic cell, which is supposed to be much more simpler than us and would have emerged much earlier than us during the early evolution of life on Earth, is still a very complicated molecule if you can look at it. It has the cell wall, the cell membrane, all the genetic material that's inside. Then there are these structures that allow it to propel called flagella. Then you have these pili that allow two cells to talk to each other. Very, very complex. How do you then imagine about this process where you go from a bunch of molecules floating around in the prebiotic soup to something that looks like this? So increasingly, the way it's thought about is that, okay, prior to that, I should tell you, these fundamental units of life, like the prokaryotes, right? I mean, prokaryotic cell, they're mind-blowing. Bacteria, right? One of the very famous examples. They have been shown to populate any every extreme niche that we can think about on our planet. You have UV resistant organisms, you have heat loving organisms or thermophiles, and some of the highest temperature tolerating organisms can tolerate up to 120 degrees C, where you and I will be burnt. Yeah, you have salt loving organisms or halophiles. You have permafrost dwelling organisms that can live at extremely low temperatures. It's unbelievable that in the later evolution of life, when you had, after you had the transition from non-life to life, you had these prokaryotes come about, and now they have pretty much populated all all possible niches on the planet where even the conditions are extreme. So then one of the things to think about is how do you actually even start thinking about this problem? So if you look at the modern tree of life, this is a very, very simplistic rendition of it. It is thought that the three domains of life as we see today, which is bacteria, archaea, and eukaryotes, can all trace their roots back to this sort of entity called the LUCA, or the last universal common ancestor. Beyond, and this last universal common ancestor is thought to have essentially come about after a lot of evolution that happened after you had the minimal entities of life come about. So over the last decade or so, this particular rendition of the tree of life has also undergone a bit of evolution, so to speak. Though not everybody subscribes to it, increasingly we are able to appreciate this because of the kind of work we do and the kind of results that we see. There's greater appreciation that you probably didn't have the single last universal common ancestor, but you had a common ancestral community of primitive cells, which did things very similar to what we see is happening to modern cells, albeit in a very, very sort of simplistic manner, just because they are not as complex as modern cells. And along the line of evolution, there were a bunch of selection pressures that sort of further constricted the number of ancestral community of primitive cells that might have been present, and then eventually led to the three domains of life as we see it today. Now, when we talk about this common ancestry of primitive cells, what do we conceive them to be? These most primitive forms of cells are referred to as protocells in the field. Minimally speaking, it is essentially a membrane-bound entity that encapsulates genetic material and or some kind of a minimal metabolism that allows it to sustain itself. So I talked to you about how we work within a working definition of life. This was ratified by a body that was put together by the NASA Astrobiology, a group of people who have been thinking about these questions for several decades now. And these people came up with a working definition of life. Life is a self-sustained chemical system capable of Darwinian evolution. So it is within this structure that we try to understand the transition from non-life to life. And within this, it is thought that very early on, there was a stage when you had these ancestral community of cells like protocells come about from the prebiotic soup. And after decades, about 6 decades of research, where are we now? We are at a place where we can draw up a story that is consistent with all the evidence that experiments from chemistry, from what we know about early Earth settings, all the geological evidence, and what biology of some of the oldest forms of life have revealed to us. So from all of these different evidences, and as you may appreciate, astrobiology is an extremely interdisciplinary area of research, where you feed off of inputs all the way from cosmology to biology and everything in between. So physics, chemistry, paleontology. There's a lot of information feeding from these various areas to try and understand how do you go from the universe, the Big Bang, to all the way to life. Now, happy to say that in 6 decades, we have actually gotten to a point where we are beginning to piece together the pieces of this puzzle. And after a century of this, okay, maybe more than 60, century of fractious effort, the story is finally coming into view. I keep saying 60, 70 years, mainly because the field of origins of life as a formal area of study happened only after a specific experiment was demonstrated in the lab. And I'll get to that in a second. So, sorry. I talked about how the field is extremely interdisciplinary, right? It feeds off of different areas of sciences. And in our lab, we basically are very interested in understanding how do you go from this prebiotic soup of molecules pertinent to extant biochemistry, how do you go from there to a minimal cell-like space, which then evolved into more more complex cells as we understand them today to be prokaryotes and eukaryotes, and essentially giving a rise to much more complex life. And in our lab, again, the kind of areas whose tools we use to understand the questions we're interested in include biochemistry, biophysics, organic chemistry, inorganic chemistry, geochemistry. So we work at the edge of geology, chemistry, and biology. And we are experimentalists, but we talk extensively to people who do modeling, who try and understand aspects from a theoretical perspective, to also bring in an understanding that is otherwise not possible only by using experimental tools. So we try to kind of get to this question from a very, very combined sort of an approach, using a very combined sort of approach. So when I mentioned the early history of life on Earth, what does it look like? This is already a 19-year-old slide, but it is something we keep going back to because it kind of gives you this beautiful overview of the main events that are thought to have happened during the emergence and evolution of life on Earth, right? About 4.5 billion years ago, you had the formation of Earth happening, and it's thought to have evolved a stable hydrosphere by about 4.2 billion years ago. And somewhere between 3.8 and 4.2 billion years ago is when this transition from prebiotic chemistry to life is thought to have happened, right? So prebiotic chemistry is the space where very interesting chemical reactions led to the formation of monomers pertinent to biochemistry as we see today. And from this space, going to this stopgap period during the early evolution of life called the RNA world is thought to have been predated by pre-RNA worlds, because going directly from this space to a biomolecule which is currently existing in our body, I can assure you, is completely non-trivial. So what is an RNA world? RNA world is a hypothesis that posits that very early on in the history of life, you basically had this one single molecule, biomolecule called RNA, doing two very important functions of life. One is encoding information, and the other thing is catalyzing reactions or doing functions. How does it happen in our cells today in modern biochemistry? Information is predominantly encoded in DNA. In most life forms, you still have forms today that exist that encode information in RNA. I'm not going to get into it now, but predominantly DNA does this role. And you have proteins, which via this important molecule called RNA, which helps in processing the information in DNA, ends up making more of itself and facilitates very important functions that allow us to basically have this conversation today. And it's thought that this is the period in the early history of life when this transition is thought to have happened. And once this came about, it was much more easier for minimal life to transition into life as we see today, where you have DNA, protein, and RNA-based life, and then subsequent diversification is thought to have happened a little after that. Sorry about that. So what are the environments that are thought to have allowed for this transition from chemistry to biology? Depending on whom you talk to in the field, there are two sort of favorite hypotheses, right? One of the hypotheses says that these reactions were facilitated in volcanic activity-driven spaces on the ocean floor. These are basically called hydrothermal vents. Here I'm showing the example of a black smoker because of the geology that is associated with the rock there and the chemistry that it supports. The gases that are released from the space are essentially what lead to this black color, and it's got a specific kind of chemistry that it facilitates. It has a certain pH, a certain temperature. This is thought to be one of the niches where these transitions have been facilitated. Even within this particular group, there is a subgroup predominantly led by Mike Russell and Nick Lane, who say that it's not the black smokers, but it's the white smokers. Black smokers are acidic vents, white smokers are alkaline vents. Again, they do not, the plume that comes out is not black in color, it's whitish in color, and it's because of the chemistry that is facilitated because of the rock's geology there. So this is sort of one group of people. The other group of people to which I belong, because to me this is much more tangible, we invoke this niche called a terrestrial geothermal pool, where you can facilitate one can facilitate, I'm not facilitating, the niche facilitates very interesting chemical and thermal fluxes that basically allow for the transition from non-life to life. Typically, you're looking at this prebiotic space made up of monomeric molecules. So let's take RNA as an example. The fundamental unit of an RNA molecule, or for that matter, even a DNA molecule, is called a nucleotide. The prebiotic space is thought to have been rife with a plethora of nucleotides, many more varieties than we see in modern chemistry now, biochemistry now. And how do you go from those monomeric space, imagine sort of individual pearls, how do you go from these individual pearl entities to a string of pearls, to a chain of pearl, which is what is the biochemical space. Our cells are made of 4 important classes of biomolecules. All of them are polymers, right? They are essentially proteins, nucleic acids, carbohydrates, and lipids. Lipids alone are not covalently linked polymers, but they are polymeric in a different sort of a way. I'm not going to get into the detail, but the other 3 biopolymers are actually polymers that result from the stringing together of the pearls to form a pearl-like chain. So these kind of niches facilitate these transitions very readily because of the inherent chemical fluxes and the thermal fluxes that can be supported there. I request Ramji Sharma to not draw these lines on the slides if possible. There are some things that are showing up on the slides. Jaya, can you prevent access to participants to be able to?
Yes, ma'am. Yes, ma'am. We are doing that.
Yeah, I'm going to hold off till this is taken care of here. Or maybe I can just move to the next slide and show you 2 other pools that can do something interesting like this. Here, these are in this particular slide, this particular niche terrestrial geothermal pool is part of this beautiful system called the Bumpus Hell. It's in Mount Lassen in California. I've been here. It's just mind blowing. You put a finger in there. When you get it out, you won't even have bones sticking. It's gone, right? It's very, very acidic in nature. So you have these kinds of terrestrial geothermal pools in other parts of the world. Kamchatka hot spring here, which is present in Russia. You have another example from the Yellowstone National Park. We do have some of these terrestrial hot springs even in India, and I will get to them towards the very end. But suffice to say that this is one of the very important geological niche that is thought to have allowed for the emergence of life on the early Earth. Now, given that we now have narrowed down on a niche which can support these transitions, what is the kind of existing model that allows us to think about this transition from the prebiotic space to a minimal life space? So if you had monomers pertinent to the formation of that membrane boundary and the formation of the genetic material that it encapsulates, all present in the same niche, these settings very readily facilitate geological events called wet-dry cycles. Wet-dry cycles are very readily facilitated by day-night cycles, by tidal activity, by temperature fluxes, so on and so forth, by sort of rains. These geologically relevant events like called wet-dry cycles, which happen even on modern Earth, are thought to have led to something very interesting. So you have you have the monomeric entities relevant to the protocell membrane and the genetic material that it encapsulates inside. When you have dehydrated conditions, you have basically these membrane molecules very readily. Membrane molecules can form higher ordered structures. This is thermodynamically driven because of the chemical nature of these particular molecules. They can form micelles, a single layered entity. They can form bilayers, which can then collapse back on themselves and form vesicles. When you have these under aqueous conditions, these monomers under aqueous conditions, they ball up to form these supramolecular structures. And these red kind of little entities are supposed to represent the monomers of the genetic molecule. If you have them in the same space and put them through a dehydration cycle, you can see that the molecules that were otherwise sort of floating around in a 3-dimensional space and trying to interact with each other with very little possibility just because of Brownian motion are now constrained in a 2-dimensional matrix, which is sort of formed by these lipid molecules collapsing on each other to form these multilamellar sandwiches. Here is an inset of a microscopy image of a real lipid molecule. So if you take these vesicles and subject them to dehydration, they can form these multilamellar sandwiches. And within these interlamellar regions, you can now have molecules interact with greater possibility, facilitating essentially these uphill reactions, which I was talking to you about. It's called polymerization. You go from a monomer to a polymer. You go from a nucleotide to a nucleic acid. So once again, on a subsequent rehydration step, if you have both of these molecules now present, your growing polymer and your you also have this lamellar lipid layers. It can bulb, it can kind of bleb back into these protocellular structures, which is what I was talking to you about. So such very, very readily plausible events are thought to have led to the formation of the first cells on the early Earth. And this inset here in this image is actually showing what we sort of demonstrate in the lab. You can start with a bunch of lipid monomers. You can subject them to dehydration. They form these multilamellar sandwiches. And if the entrapped molecules within them are fluorescent molecules, once they rehydrate, you can actually sort of see these things bleb out into this cell-like or tubular structures. And it's thought that selection that acted on these populations essentially led to their evolution and to better sustained populations. And these cycles, multiple cycles, DHRH cycles followed by selection, are thought to have allowed for this transition from this prebiotic soup to the minimal life space. Oparin and Haldane theory was one of the oldest theories that talked about how do you go from this inorganic space to an organic space. What both of them told, and back then no internet, right? Forget internet. There wasn't, basically internet. There wasn't even low speed internet back then. We're talking about around 1940s. Oparin is a Russian scientist. Haldane is got a very interesting connection to India, to West Bengal and to Pondicherry. Both of them separately posited that if you had a prebiotic ocean composed of molecules, inorganic molecules like methylene dicarbon, water is sort of the backdrop in which all of this drama happens. No water, no life, at least not that we know of to date. Carbon dioxide, ammonia, you had all these molecules. And if you had an energy source like lightning or ultraviolet radiation from the sun, you could have, in principle, facilitated the spontaneous formation of these organic molecules. Here it's sort of a representation of a little peptide, but essentially you can go from the inorganic molecule space to the organic molecule space. Haldane went on to coin the term the primordial soup. This was a theory that two people independently posited, two of them who did not know of each other's existence. And this was around late 1940s. People started talking extensively about this in research circles and science circles. And before you know, a young gentleman then, his name is Stanley Miller, went to Harold Urey and basically said, I want to try out this experiment, because a hypothesis is a hypothesis till you sort of show beyond doubt that it is plausible, right? Or it is possible. So what they went on to do is put together this contraption. This is the original contraption, yeah? Here is your flask where you can hold the prebiotic ocean, which has all the ingredients in it, methane, ammonia, and dihydrogen and water. You have temperatures are thought to have been very hot on the early Earth. So you have a burner down there, which essentially allows you to heat up this prebiotic ocean ingredients. You can basically have water evaporate and carry with it some of these molecules. And it goes into this chamber where you have an electrode setup that can basically provide a very high source of energy to the molecules that are present here. And they interact to form a lot of other molecules. Then the vapor comes down. You can use a condenser to once again liquefy the gas, and it comes down here. You can collect it and you can take out samples for analysis. And they allowed the cycle to keep on going, right? They collected samples over multiple time scales. And what they showed for the first time using experiment, that you can go from a mix of these inorganic molecules to a mix of these organic molecules. And back then they didn't have really high-end techniques to show this, but they had some very ingenious biochemical tools that allowed them to actually show beyond doubt that you could have these molecules form. What is very interesting, and this is a small trivia for those interested in these kinds of trivia, Stanley Miller's first student was somebody called Jeff Bada. And one of his last but one students, Jim Henderson Cleaves, a very dear friend of mine, the two of them got together about a decade ago, and Stanley is supposed to have been extremely anal about how he kept his notes, how he saved the samples from various iterations of this experiment. So what they did is they retrieved some of these samples after very carefully going through his notes and understanding which vial had which reaction, so on and so forth. And they used some of the very high-end modern techniques to actually show that it was not just the set of molecules that they showed as plausible, organic molecules that they showed as plausible under these conditions. The modern high-end techniques that can determine parts per billion amounts of molecules actually went on to show that you can have even more complex amino acids come about from this kind of a reaction. Here, you only see glycine, glutamic acid, alanine, but you can have the more complex molecules which have these hydrophobic heterocyclic rings like tryptophan, tyrosine also forming in these conditions. So it was the first proof of principle that came about, which basically showed that the Oparin-Haldane hypothesis was indeed valid. This set the stage for basically a field of research called the origins of life research, mostly prebiotic chemistry, because this is the chemistry that predated life. This was the famous year 1953, which was also the year when the double helix structure was basically described. So suffice to say that in these decades since 1953, there has been enough research coming out from colleagues' labs who do very, very interesting prebiotic chemistry that you can go from a mix of these inorganic molecules that are shown here to result in precursors that eventually lead to molecules that result in membrane structures, molecules that result in proteins, these are amino acid precursors, and nucleotide precursors that can result in formation of DNA and RNA, which is actually the genetic material. So what is thought to have happened subsequent to the emergence of protocells is basically something, I mean, a lot of events must have happened that, and a few different selection pressures might have come into play. We do not understand too much about all these things, which essentially led to the transition from protocells via a community of ancestral cells to what we see more today. This is cyanobacteria. It's one of the oldest prokaryotes that is thought to have emerged on the early Earth, and now the evidence, earliest evidence goes back to about 3.85 billion years. And it's super cool to even think that you can use modern technology to try and understand biosignatures from samples, rock samples that are so super ancient. So where are these prokaryotes thought to have originated? Yeah, they could have originated anywhere where the ingredients for life was rife, particularly in the kind of niches that I talked to you about. Here is one of the very important examples. These are that allowed us to kind of show the presence of these cyanobacteria all the way back to 3.5 billion years ago. Now, like I said, we have evidence that dates it back to 3.85 billion years ago. Here is what these pillow-like stone structures are called: stromatolites. This is basically in Perth, which is in Western Australia. You can basically see that, I think this is Shark Bay, if I'm not wrong. Basically, you can see that these pillow-like rock formations, when you take one of these samples and you slice it, you see these alternate layers of what looks like gold and black regions. Fundamentally, it is thought to have resulted from the interaction between cyanobacteria via some jelly matrix, which also entrapped sediment and rock material that resulted in these kinds of structures. These kinds of structures grow at about 1 millimeter per year. Yeah, you can carbon date them and stuff, and they basically showed that this is one of the oldest geological evidence that is out there, now much older. But importantly, these organisms are what are thought to have eventually led to the oxygenation of the earth, because very early on in life, when we think about the transition from chemistry to biology, it was all under anoxic conditions, no oxygen. But subsequent to the emergence of these prokaryotes, you had this very important event called oxygenation, which essentially is thought to have led to dramatically changing the organisms that essentially survived and evolved on the earth subsequent to this process. Yeah, why is oxygen very important? It drives one of the most important life events on earth called photosynthesis, where these prokaryotic organisms can use the water and the carbon dioxide in the atmosphere, yeah, to cook up, synthesize food, and oxygen is literally released as a byproduct. They use solar energy as the light, energy source. And the byproduct of their photosynthesis process is essentially oxygen. And all of the oxygen that you see on Earth's atmosphere today is thought to have resulted from this process, which was facilitated by organisms that evolved about 3.5 to 3.8 billion years ago. Now, when did our ancestors evolve? It's thought to have happened about 2 to 2.5 billion years ago. This is when eukaryotes came about and the first multicellular life is thought to have come about. And how do you think, how does one envisage that this happened? This basically started happening when molecules from when organisms from two different domains of life came together and started interacting with each other and led to the formation of this third domain. It's thought that archaea and eukarya, prokaryotes came together and resulted in the eukaryotes. Yeah, but this is what we are. We belong to this kind of group of organisms. And here is an example of a choanoflagellate roset colony where you have a bunch of cells coming together and interacting, resulting in these rosets. Sorry about that. Yeah. So what is it that we do in our lab here? Just to give you a flavor of how do we even start asking these questions in a much more systematic manner? We try to understand how do you go from the prebiotic soup to minimal life by basically asking these sets of questions. First is, we want to understand what is the kind of prebiotic chemistry that led to the formation of the building blocks of life. And let's assume that you could form, and there's no reason to assume, there's enough data evidence out there, the experimental evidence out there from colleagues' lab that have shown that you can readily form these amino acids and these nucleotides in under early Earth conditions. So we are very interested in understanding how do you go from that monomeric space to a polymeric space when there were no enzymes. How do your cells and my cells make these DNA and RNA? They use these extremely highly precise, highly engineered molecules called proteins. Now, then the question is, where did the proteins come on the early Earth? It becomes a chicken and egg problem. Which came first? Because you need the information that is encoded in the nucleic acid to make a protein. You need a protein to make the nucleic acid. So, you see that chicken and egg problem. This is where the RNA world hypothesis that I told you about, which is a very heavily accepted hypothesis in the field. It's not the only one out there, but it is very attractive for a multitude of reasons. Basically provides a relief from this chicken and egg problem. Here is a molecule now, an RNA called RNA, which can readily form only by chemical means. We do not have to invoke biology or heavy duty, highly engineered enzymes. So we in the lab try to understand how do you go from the nucleotide space to an RNA space via non-enzymatic polymerization mechanisms. We also want to understand that once you come about with interesting functional length RNA molecules, because I told you RNA can also function as enzymes, it's very important to understand mechanisms that might have allowed for faithful replication of such long polymers, information in such long polymers. And it's a completely non-trivial problem. Again, in your cells and my cells, highly evolved engines, machines facilitate this replication mechanism from DNA to make more copies of DNA. But again, proteins didn't exist. So what are the mechanisms which are non-enzymatic in nature that allowed for reasonably faithful replication of these the information in these molecules? And then what kind of crosstalk happened between the RNA world molecules and the membrane compartments which form the outer boundary of the cells to eventually lead to the emergence of protocells? And we are interested in a lot of other things. Just to sort of, can somebody tell me the time? 5:44. Can I take 5 more minutes?
Yes, ma'am. Sure.
Yeah. So then maybe I can just quickly tell you about how we are interested in looking at multiple aspects of astrobiology. We are interested in looking at origins of polymers, biopolymers, fundamentally informational polymers, and also want to understand how protocells, the earliest of protocells, emerged under early Earth conditions. We're very interested, as I said, in understanding information transfer by non-enzymatic means. We like to understand the rates, the kinetics of the process. We like to understand the error rates in the system. We're also very interested in understanding prebiotic catalysis that was facilitated on the early Earth by molecules that were way more simpler than your enzymes, right? And one of the ways that we think about in this field is that this might have been facilitated by metals. So we look at metal-mediated inorganic catalysis. We also look at crosstalk between some of these metals and scaffolds like porphyrins, which are found in some of the most ancient enzymes on Earth, to look at minimal catalytic systems that can facilitate interesting functions. And all of the work that we do a lot of experiments. We recently donned, about 5 years ago, donned the hat of becoming a field scientist, so to speak. We are still amateurs, though. We are also very interested in trying to understand how both lab simulated conditions and analog environment studies, where we do experiments both in the field and bring back samples and work with the realistic samples back in our labs. We kind of want to understand how we can eventually understand important prebiotic processes that allowed for this transition from chemistry to biology, because it has very important habitability implications. If you tomorrow send a sort of orbiter to Venus or to, say, Europa, what are the kind of signatures is it supposed to look for? So many of the processes that we study are very, very complex and involve the formation of a lot of intermediary molecules. And even those molecules, the nature of those molecules, allow us to try and get a sense of the kind of chemistry that can be facilitated in exo-Earth planets, right? Here's one very grand interest where we like to understand how do you go from the prebiotic soup space to the pre-RNA world space to the RNA world space. This work was started by my first student, Chaitanya, and it's still being continued by a student called Shikha, and this organic chemist postdoc that I have, who's a nucleic acid, synthetic nucleic acid chemist. He's basically trying to help us come up with synthetic nucleic acids to try and understand this pre-RNA world space a little better, and it's been really fascinating. In the other project, we're very interested in understanding how replication might have been facilitated in a very complex prebiotic soup. The soup that I'm talking about is the result of interaction of a lot of different kinds of inorganic molecules. So it can basically result in a solution where you have a lot of different kinds of organic molecules. So when we start thinking about some of the fundamental processes involved in prebiotic chemistry, we are completely ignoring the heterogeneity in the prebiotic soup. So in one particular project in the lab, we kind of make our lives a little more hellish, and we stop and account for the prebiotic heterogeneity in this prebiotic soup, albeit in an incremental sort of way, to try and understand how could you have used the information in a template nucleic acid strand to extend a primer to make an intact new replicate strand. So this is sort of the paradigm with which we work in, but there are other few paradigms that exist, not going to get into the details. But Gauri, who's now with us, has gone on to sort of take this project further after Nirja started it. And we are very kind of systematically showing that in the presence of this heterogeneity in the prebiotic soup, your mutation rate during information transfer actually becomes exaggerated. Is that good? Is that bad? I will not talk about it, but that's for you to think about. In another project, Manish, a grad student, a senior grad student, and also Anupam are very interested in understanding the crosstalk between the membranes and the informational and the catalytic polymer. And very recently, Manish showed that if you had both these molecules, the amino acid and the lipid monomer present in the same prebiotic soup, and when they are subjected to multiple wet-dry cycles, you can actually form a new class of molecules which nobody talked about before in a prebiotic context. They are called N-acyl amino acids. Essentially, they form as a result of reaction between a phospholipid and an amino acid, and they can do something very cool. They themselves can assemble into these compartments called vesicles very readily under conditions where whatever we worked with till now don't readily form membranes. So that's very, very cool, because we are still not clear where these interesting reactions happen, because some were facilitated under acidic conditions, some under neutral, some under basic. So where did these transitions from non-life to life happen? It would have happened because of result of events happening under a variety of range of pHs. So it'll be very interesting to actually find a whole suite of molecules, or describe a whole suite of molecules that might have been present in the heterogeneous prebiotic soup, but we still don't know of them, and to try and systematically understand how might they have formed these compartmental entities to form resultant protocells. Here's Sushoman, and this is Shoradeep, a senior grad student and a junior research fellow right now. Both of them are very fascinated by the question, how did the prebiotic landscape of amphiphiles, sort of what are the kinds of selection pressures that acted on them? When I say selection pressures, what are the most obvious selection pressures you can think about? They are selection pressures, that evolutionary pressures that even our cells are undergoing. pH, temperature fluctuations, presence of cations. Cations are very important in formation of these membranes, but there is the sweet spot. Too little, membranes don't form. Too much, membranes collapse. But the same cation, magnesium, is very important for the functioning of the genetic polymer. It's very important for these RNA enzymes called ribozymes to do their function. So we're very interested in understanding the trade-off of what are the entities that survived when you play around with many of these parameters. And they're particularly interested in understanding the emergence of the primitive cellular membranes or protocellular membranes. And there is a notion of how it might have happened, and I'll not get into details unless somebody is interested. And finally, Shikha very, very interested in understanding how these prebiotic catalysts emerge, right? Now we have these extremely complex highly engineered molecules called enzymes, but it started very early on. It was just metal ions doing some of this. So there is this sort of pathway that we can think about where these metal ions would have sort of done this coordination with some scaffolds to form metal scaffold complexes, which then interacted with short peptides and stuff. And over these billions of years, you see enzymes, but very early on, it must have been these various entities at these various levels of complexities that facilitated catalysis. And this is something that she does. Like I said, we also we've also started donning the hat of field researchers. This is an expedition to Ladakh, which happened in 2016 under the aegis of Mars Society Australia. And we had collaborators from NASA join us. So it was BSIP, Birbal Sahni Institute of Paleosciences, and ISER Pune that was involved in this expedition on the Indian side. And we basically went and visited Ladakh. Ladakh is this fantastic Mars analog. It has a lot of these geological features that you can find on Mars. So what this means is sites like Ladakh, very high altitude deserts, right, where the environment is very thin. Sites like Atacama in Chile, the Atacama Desert, are all fantastic field sites to do analog studies to better understand how the reactions that you're interested in actually pan out under realistic conditions. So that is what we are doing now increasingly in our lab too. And just to give you sort of a flavor of where interesting astrobiology research happens in the country, of course, at ISER Pune, but you have other players too. You have Birbal Sahni Institute of Paleosciences, where there are a lot of paleontologists that are trying to understand the paleontological angle. And there's a bunch of groups there that are also trying to understand some of the extreme life that came about, cellular life that came about on the early Earth. Then you have colleagues from Indian Institute of Astrophysics who work on some of the astrophysical aspects. Then you have colleagues from Physical Research Laboratory who are also working on planet formation, which is extremely important to understand if you want to understand why certain planets went on to harbor life versus certain others that did not. And more recently, a very dear friend has started the Center of Excellence in Astrobiology, Amity Center of Excellence in Astrobiology in Vashi campus. And yeah, a lot of fun times for astrobiology research in India. And here I'll just finish off with giving you a sense of where astrobiology is today. Astrobiology today is embodied in this Perseverance rover, right? What is it doing? The Mars 2020 Perseverance rover is actually looking for signatures of life on Mars, which is thought to have harbored conditions very, very early on, which look a lot like modern Earth conditions, right? Or conditions, yeah, modern Earth conditions. So I will just leave you with this thought by saying that it's a very complex problem. It's completely non-trivial to factor in all the parameters that might have come into play. We can break it down into bite-sized chunks and ask questions that will slowly allow us to sort of fill in this very grand puzzle at some point. So it's a very knotty problem, but it's not insoluble. Yeah. Thank you for your time, and I appreciate your patience. Sorry. Yeah. I'm ready to take questions.
Yes, ma'am. Thank you for such a wonderful talk. It was an eye-opener for me, and I really hope that the participants felt the same way. We can now take some questions, and the participants can ask your doubts by raising your hand or even in the chat box.
Maybe I can stop sharing, right? So we can.
Yes, ma'am.
Something. Yeah. Ma'am, your voice is quite low. Jessing, I'm hearing it only now. Jay, did you have a problem listening to me?
No, ma'am. I think it was fine. It was audible.
I'm going to put the ball in your court, Jessing. I think you had an issue at your end. I hope not everybody had this issue because it seems like it only came from him. Yeah, happy to take any questions. No question is trivial and all questions don't have answers. So that's the paradigm. Nikhil, you want to say something?
Ma'am, I have a doubt. How scientifically valid is the panspermia theory? Is there any scientific scope for it, or is it just a hoax?
This goes back to a statement I made, right? A hypothesis is a hypothesis till proved otherwise. I think there are a bunch of people, a small handful of people across the planet, including some from India, who still are trying to systematically understand the panspermia hypothesis. If you put a gun to my head and say, Sudha, you can say only yes or no. Do you support it or no? My answer is no. I do not, because I find it really difficult. I am very comfortable with thinking about or envisaging how molecules pertinent to extant life and many others, which we don't see life using, could have very readily ridden extraplanetary objects. But to imagine actual cells, actual biological entities riding these exoplanetary bodies and then surviving the transit. First of all, how do they get into the core of this comet is a question. Then the re-entry into the Earth's atmosphere is completely non-trivial, right? So how do they survive it? It just boggles my mind. So my answer is valid hypothesis, reasonable. Valid, no. But should more work be done on it? I feel like doing more work on extremophiles, trying to understand how highly UV resistant extremophiles, right, survive, will help us better understand sort of how does the re-entry get facilitated. But I'm not going to buy it till I see better results. Yeah. Any specific observation which has given rise to such a possibility? Nachiket, Nikhil, I'll get to you in a second. Nachiket, your meteorites, right? Meteorites have ice crystals all over. Water forms very readily because of interactions in the interstellar medium, just like many other molecules are formed in the interstellar medium. And when you have planet formation happen, and I do not know the nitty-gritty details about the process, but suffice to say, planetary science colleagues have actually shown that you can have water crystals actually getting trapped into the core of the meteors. So when you have this meteoritic entry, I told you that it has to survive the re-entry process, which is non-trivial. It's going to burn out all the outer layers. However, you have meteorites that are several kilometers huge. And I'm talking about a period in very early history of life, around 3.8 to 3.9 billion years ago, where it's supposed to have been the late heavy bombardment period, where for several millions of years, you had these planetary entities constantly bombarding Earth and Moon. Enough of these things have bombarded. So even if a good bit of it got destroyed during the re-entry, you still have enough of these ice crystals in the core of the meteorite that survived. And once the temperatures on the early Earth went from crazy hot to reasonable, you could have had these things condensed to form water. It is thought that all the water that is present on the planet today actually finds its roots back to the crystals in the meteorites. I'm going to go to Nikhil. He had his hand up, and then I'll get back to two other questions in the chat window. Nikhil, you want to ask what you want to ask? Nikhil? Was Nikhil the one who asked the first question? Kaushik, he was the one. No, there was somebody else whose hand was up. Anyways, I'm going to take Shubham's question. Can you explain more the RNA world hypothesis? The RNA world hypothesis, Shubham, basically says that during the very early history of the transition from non-life to life on Earth, you had much more simpler entities, complexity wise. You and I are ultra complex. In comparison to us, a bacteria is way less complex. And we are talking about a time on Earth which is even further back in the early history of life, when it's thought that you actually went from very simple organic biomolecules to more complex biomolecules like your nucleic acids. And RNA world basically suggests that you did not see right now, if you want to think about life on modern Earth, right, you have to think about how 4 different biomolecules came about. We already know about how lipids would have come about. Because of this exogenous delivery, monomers are thought to have been very readily delivered to early Earth. And there are also something called Fischer-Tropsch type synthesis mechanisms that are facilitated on terrestrial atmosphere that can form these amphiphilic molecules. Lipids are amphiphilic molecules. They have a water-loving head group and a water-hating chain. So we know that your prebiotic soup would have had the monomers of the lipids, the monomers of the nucleic acids. If you can think of a way to sort of oligomerize the nucleotides to RNA, you now have made a molecule that can do 2 very important functions of life. You don't have to figure out how all of them came about. You just need to figure out how RNA came about, because it can also encode information, yeah, of life, and it can facilitate functions like protein enzymes. RNA enzymes are called ribozymes. So it kind of simplifies the problem of understanding this transition from non-life to life. And hence this RNA world hypothesis is very interesting. And importantly, around 80s, one of the first proof of principles for this particular hypothesis to be valid came about when Tom Cech and his group demonstrated the presence of ribozymes in certain living entities. And also the most important reason why this RNA world hypothesis gained a foothold even in the 50s and 60s is because of the fact that the core of your ribosome, which is the heavy duty machinery that forms the protein molecules, the core of the ribosome, which brings two amino acids together, uses the information in the nucleic acid in your, basically you have to transfer the information from DNA to an RNA. It's called, the process is called transcription, after which the information in the RNA is used to make a protein. The core of the ribosome is actually a ribozyme, where the fundamental addition reaction, condensation reaction of two amino acids to form a peptide bond is facilitated by an RNA molecule and not a peptide molecule. The protein surrounding the RNA is very crucial to stabilize the RNA in a certain structure, which facilitates this catalysis. So this was sort of the first real big proof that, oh yeah, maybe the RNA world was indeed a very important stopgap. Now, over the last 2 decades, it's become very clear that you can't go directly from the prebiotic soup to an RNA world. It's a completely non-trivial route. So maybe it is thought that maybe this RNA world was predated by pre-RNA worlds where you had simpler but related nucleic acid molecules that much more readily assembled from the prebiotic soup and later on evolved to the RNA and DNA world that we see today. Yeah. Satyam says, is it possible that somewhere out there, there would be some different life forms which do not require the conditions that we require as for finding life in other planets? Yeah, so Satyam, what we understand about all of the biochemistry that's possible on Earth, I mean, look at the tree of life. Every single entity, all the way back to the base of the tree of life, slightly above Luca, where we have kind of described a lot of these organisms, all use the exact same set of molecules, right? And these exact same set of molecules, DNA, RNA, and protein, can do what they do only in the context of water. These three molecules have a lot of charges on them. So they basically, when they try to come together, because of these charges, they can repel each other. The only reason they're able to even come together and do something is because of water providing that canvas. It allows these molecules to stay dissolved so that they can interact. Even there you have all these charge related drama happening. I'm not going to get into it, but water is a universal solvent. I do not know of too many things that don't readily dissolve in water other than organic solvents. It can dissolve a ton of things because of its very high dielectric constant. So I'm not sure if there is anything else out there that doesn't look like life as we understand, but if there are completely, if the Earth history, if the, sorry, planet formation history is completely different, and if they have, see, all the elements are the same. The interstellar medium forms a ton of elements and some molecules. All of that chemistry is universal, literally. So I don't know what else can behave as mind-blowingly as carbon. Silicon is a close second, but it is a very distant close second. So I'm not sure if there are other kinds of life forms out there. Fundamental biochemistry might be the same, but how they evolved might be different. The selection pressures might have been different. So when we say different, they might end up looking a little different, not like the aliens, but I'm talking about when we say life out there, it's much more simpler to imagine how the simpler life came about, like bacteria, right? So we already, there is this term called microbial dark matter. What we understand about organisms on our planet is still a tip of the iceberg. We've only scratched the tip of the iceberg. We still do not know about a ton of them. For example, if you bring back samples from these hydrothermal vents, saying, see, hydrothermal vents are the black smokers that I showed you, are rife with life. You find these weird white lobster-looking things, crab. They're called some white crabs or something. You have really evolved complex organisms that are surviving on the ocean floor, where that pressure will make you and me burst in like a handful of milliseconds, right? So even if you want to think about the chemoautotrophs that are there and try and simulate conditions to whatever extent we understand in the labs to culture them. We can't even culture them. It's non-trivial. So we don't even understand the microbial matter that exists on Earth to be able to remotely start thinking about these other contingencies that life might have used. Maybe there are examples out here itself and we still do not know. Yeah, this is a long-winded answer, but that is what I've understood from all the reading that I've done and all the talks I've heard in this regard from colleagues in the field. Man, will Mars also have same life that Earth had over billions of years and same evolution? The geological history of the planet, Ayush, there seems to be a lot of similarity in the geological history. Right now, Mars is not active, in the geologically inactive, in the sense there are no volcanoes. But very early on, it's thought that in Mars's ancient history, there was volcanic activity. And there's a lot of important understanding that's come from understanding how plate tectonics are crucial for life's emergence. So if we are looking for life on Mars, which is what Perseverance is doing, or for signatures of life on Mars, I would imagine it is very, very close to what we see on Earth today. Yeah. Swati says, is it mandatory that the source and the conditions of life remains the same as in Earth? Can there be possibilities that conditions may differ for evolution of life? Yeah, so it can vary. Yeah, it can vary. Even now, these selection pressures that I talked to you about are essentially environmental parameters that can act on an interacting set of molecules and then can basically allow some to win out and some to lose out. Yeah, basically survival of the fittest kind of a scenario, right? We can systematically do experiments in the lab to at least set a bound, upper and the lower bound, of where we can look for interesting reaction possibilities and interesting outcomes of such reaction possibilities on other sort of planets within our solar system and outside of our solar system. We can kind of try to think about these bounds and work with it. Is it mandatory? I would say no, but will we see one and say, yes, that is it, but that's not the same as, that didn't evolve under the same conditions as our life, yeah? That would be much more non-trivial. Having said that, there's a whole bunch of research that is happening in trying to also factor in abiotic signatures. A lot of the reactions that we do actually allow us to generate a library of abiotic molecules that are not considered lifelike, yeah? But they are very closely related to life. These so-called alternate nucleic acids. Our cells basically use only a subset of these nitrogenous bases on our nucleic acids. You have adenine, guanine, thymine, cytosine, ATGC, and in RNA you have uracil, and you have some modifications that are possible on these five kinds of nucleotides to form certain other variations of this, so to speak. But that sort of does, knowing that they exist tells us two things. One, the possibility of having other kinds of these bases. Fundamentally, your nucleotide has three units: the nitrogenous base, the sugar, and the phosphate. Yeah, nitrogenous base is what gives the identity to the monomeric unit, because the sugar and phosphate are the same on ATGC and U and the other variations thereof, right? But what this tells us, that the fact that 5 of these exist and some variations of that also exist, tells us that maybe very early during the early history of life on Earth, maybe there were way more of these molecules that were plausible under the early Earth conditions. And actually, there's a very dear colleague in the field who has done a pen and paper exercise to put together all the different kinds of heterocycles that can be possible, 6-membered and 9-membered rings. Systematically, we've been able to rule out a subset of them which are not plausible under early Earth conditions. Some have been found in meteorites. So yes, check, we can still consider them. So there are ways to think about this. And maybe there are ways to understand if you can detect some of these non-life signatures on other planets in our own solar system or on exoplanets, and then say, maybe this particular planet can potentially, would have potentially harbored ancient life, or can potentially harbor life that is yet to come about. So complex situation. Ma'am, what's your thoughts about silicon-based life forms? Yeah, like I said, silicon is not carbon. Silicon is a very, very, very poor version of carbon. Carbon can do some amazing number of under a different set of conditions, does silicon become very active? I do not think so, because at least the way, the fundamental pathways by which these elements are formed in the interstellar medium are very similar. So I do not know if the geology or the geochemistry of a certain planet will suddenly dramatically change the way the silicon atom will interact in that setting. My answer is the possibility of such life forms is not great. It might be possible on Earth because of our own intervention and tinkering, right? Artificial intelligence, but I'm not really sure how readily they might be a part of life forms that existed on a completely different planet. Ma'am, tardigrades are kind of immortal organisms. So what if their genetic material is transferred to other sort of organisms? Can they gain that strength? Yeah, so it's not the genetic material that makes tardigrade badass. It's actually a sugar that its cells contain. Tardigrade is made of the exact same genetic material that you and I are made of. DNA, RNA, nothing different. Same sets of bases on the DNA and RNA. So everything is just the same. What is different is there is a very interesting sugar in its cells called trehalose. And trehalose forms this very robust matrix kind of material, which ends up forming when you subject the molecule to, when you subject the organism to extreme conditions, right? Dehydration, extreme dehydration, very high UV radiation. Trehalose ends up forming this very robust and hard matrix, which actually provides the, which actually protects the other molecules in the cell, like your nucleic acids. So that is what makes tardigrades badass. So yeah, it's not about their genetic material. Maybe if we learn how to use trehalose as our sugar molecule, what is the sugar molecule in our body that we use mainly? What is the
Glucose.
okay. Sorry, ma. Somebody said something, and you were right. I just couldn't hear the full thing.
Yeah, glucose. Like glucose.
Glucose is the one of the most famous monomers among the family of sugars that our cells use. There are a few others also, yeah. But having said that, the polymer of glucose that is used as an energy source in our body is called glycogen, yeah, which is stored in the liver. Glycogen is not like trehalose. Trehalose is anyways a monomeric entity. So yeah, if we figure out a way to use trehalose for our fundamental energy source, maybe we might become tardigrades. I don't know. I have no clue. That's something to think about. Ma'am, since Mars has a cooler temperature, is it that formation of life is not possible as it requires? Ah, that's an interesting question. This whole thing about biologically relevant monomers having to polymerize to form biopolymers being a very important process, the thing that I kept harping about essentially is a condensation reaction. What is a condensation reaction? You bring two molecules together. A water molecule gets kicked out and the two monomers now get connected by a covalent linkage to form a dimer. And then you can imagine this process going on. Your nucleic acids, they are the result of a condensation reaction in which a phosphodiester's bond is formed between two nucleotides. In an amino acid, the condensation reaction results in the formation of a peptide bond. When I say a condensation reaction, and when I say that you're trying to kick out a water molecule, when can that happen? When you invoke very high temperatures, right? When does water start to vaporize? At 100 degrees C. And hence a lot of the, and when you invoke these terrestrial geothermal niches, they are very high in temperatures. Even modern, even these modern hot springs that you come across, some of whose images I showed you, are anywhere in the regime of 55 and above. And some of the ones we work with are at 85 degrees and 90 degrees. And hence it's much more easy to envisage that high temperatures, easier to kick out water. However, that is not the only one means of condensation. Turns out, if you take water, add salt to it, like sodium chloride, yeah, a very interesting thing happens. at a certain strength of sodium chloride and above, your freezing temperature of water goes down. You get into this you have this phenomenon called eutectic phenomenon that occurs, where you can actually now result in a scenario where you have crystalline ice, and because you started with salt in that, you have these brine channels where the salt gets concentrated. Now imagine a scenario where you have monomers of these nucleic acids called nucleotides present in the starting reaction mixture, where you basically started with a buffer / water, had the monomers of nucleic acids and salt, and you let basically it freeze. In these brine channels, you can have condensation happen. And how is the condensation happening? Salt loves water. Salt clings on to water and takes it away from the interacting molecules. So you can actually facilitate uphill polymerization reactions even under eutectic conditions where you have frozen ice. So the question is, can you find nucleic acid-like molecules at cooler temperatures? The answer is yes. And this brings me to one very important aspect of looking for life outside of Earth, right? There are 4 very famous bodies in our solar system, which are front runners for life detection. One is Mars. The other 3 are actually moons. Yeah. One is moon of Saturn called Titan. One is the moon of Jupiter called Europa. One is the moon of Enceladus. Enceladus is the moon of also Saturn, if I'm not wrong. Yeah. So these moons are essentially what you called ice, yeah, ice laden icy worlds. They are kind of ocean worlds, ice laden ocean worlds. There's an ocean on their atmosphere, on their surface. For example, if I'm not wrong, Titan is one of those rare moons in our solar system which is very dense, which has an atmosphere which is very dense. So you have water on its surface, you have an ice layer covering completely the outside of the body, the planetary body. And then there is a notion that you also, there's actually been proof that you have hydrothermal activity in both on Enceladus and Europa. So what this all is indicating is you have geological conditions that might have been able to either host ancient life or also hosts extant life. So you can still very much look for these signatures, biosignatures on planetary bodies which are not necessarily hot or are not as hot as how early Earth was. You can have some of these processes even facilitated under much, much cooler temperatures. But you need to have, see, the fact that your these ice-covered ocean worlds actually have volcanic activity means that there are niches in there where you have very high temperatures. There's volcanic activity, right? You have plumes, hydrothermal activity. You have plumes coming out, which flybys have collected and studied. So you need to have geological settings where you have some high temperature niches and some low temperature niches to facilitate multiple of the processes to eventually sort of lead to a space where you then had boom, life. So yeah, sometimes it becomes very difficult to wrap your head around all of these things. You say you need high temperature for this, but you need cooler temperature for this. So people kind of invoke scenarios even present on modern Earth today where you can have multiple of these happening simultaneously. You have this amazing gradient when you talk of hydrothermal vents, right? You have a pH gradient, you have a temperature gradient, because from the core of the Earth, you're at extremely high temperatures, but by the time you come and hit the ocean floor, you're at extremely cold temperatures. So you have this whole temperature gradient where different temperature layers facilitate different reactions. And hence the hydrothermal vent hypothesis people are like super sure that hydrothermal vents were where life emerged. But let's assume that happened. My problem is to understand how do you prevent the problem of dilution as soon as your life forms formed and came out of the vent systems. It is the vast ocean. I don't know. Very hard to, I can't think about how that might have been possible. So yeah, again, a long-winded answer. if that is about it, Jaya, can we wind up for the day? I will take Ronak's question. That'll be the last question. Yeah. So you can actually, Ronak, look for two kinds of different things, right? You can look for organic molecules and you can look for biomolecules. Many of these organic signatures, including your hydroxy acids, are very readily formed under certain set of conditions. And you can use these infrared kind of infrared containing telescopes to actually look for chemical signatures that can tell you what are the molecules that, and kind of work your way back and try and understand what are the molecules that might be present there that are resulting in these signatures. So you can look both for organic signatures of organic molecules, and you can also look for signatures of actual biomolecules as we see in extant biochemistry on Earth. So you can do two kinds of things. And if you look at some of these IR spectra that are generated from these IR IR telescopes, I'm sorry, it is crazy. It is so complex. We just understand, again, tip of the iceberg. Yeah, so a lot of things are possible. We just don't understand all of it yet. Yeah, Jaya.
Yes, ma'am. We would like to thank everyone for their active participation, and thank you, ma'am, for taking some time to do this.
Thank you. Thank you for having me. It was fun.
Yes, ma'am. Thank you, everyone, for joining, and have a great day.
Thank you. Ciao. Bye.
Yes, ma'am. Bye.